Hello, my name is Michelle King, and today I will be presenting a PowerPoint presentation on inheritance. All the information in these slides has been generated by AI, ChatGPT to be specific. So let's cover epigenetics. Epigenetics is a field of biology that explores changes in gene expression or cellular phenotype that do not involve alterations to the underlying DNA sequence. Instead, epigenetic modifications can influence how genes are turned on and off, and they can be heritable across cell divisions and, in some cases, across generations. Epigenetics plays a crucial role in development, cellular differentiation, disease, and evolution. So let's talk about our epigenetic modifications. So let's start with DNA methylation. This is one of the most well-known epigenetic modifications. It involves adding a methyl group to a cytosine base of DNA, typically at the CPG sites, regions where a cytosine is followed by a guanine. DNA methylation often leads to gene silencing and is associated with stable, long-term gene expression patterns. Second, let's talk about histone modifications. Histones are proteins that package DNA into chromatin. Chemical modifications to histones such as acetylation, methylation, phosphorylation, and ubiquitination can alter the accessibility of DNA. For example, histone acetylation is generally associated with gene activation, while histone methylation can have different effects depending on which amino acid residue is modified and the extent of the methylation. Lastly, let's touch on non-coding RNAs. Non-coding RNAs, such as microRNAs and long-coding RNAs, can influence gene expression post-transcriptionally. MicroRNAs can degrade or inhibit the translation of a messenger RNA or of our L and C RNAs. This can interact with chromatin and influence gene regulation. Now let's move on into sex chromosomes. Sex chromosomes are a pair of chromosomes that determine an individual's sex or gender. In most mammals, including humans, sex chromosomes are denoted as X and Y. These chromosomes carry genes that control the development of sexual characteristics and reproductive systems. Chromosomal composition. Humans have 23 pairs of chromosomes, with one pair being sex chromosomes. Females have 2 chromosomes, denoted XX, while males have one X and one Y chromosome, denoted XY. The X chromosome is larger and carries numerous genes related to sex determination, while the Y chromosome is smaller and contains fewer genes. The presence or absence of the Y chromosome determines an individual's sex. If an individual has 2 X chromosomes, they develop as female, while an individual with one X and one Y chromosome develops as male. The SRY, sex determining region Y gene, located on the Y chromosome, is the primary genetic factor responsible for male development. It triggers the development of testes, which can produce male hormones, for example, testosterone, leading to the formation of male reproductive organs and secondary sexual characteristics. Let's talk about inheritance next. So, males inherit their X chromosome from their mother and their Y chromosome from their father. Females inherit one X chromosome from each parent. Since males have only one X chromosome, they can pass on only one X chromosome to their offspring. Females, on the other hand, can pass either of their X chromosome to their offsprings. So let's dive right into sex-linked inheritance. Genes located on the sex chromosomes are referred to as sex-linked genes. Since males have only one X chromosome, they are more susceptible to disorders caused by recessive X-linked mutations. Examples of X-linked mutations include hemophilia, color blindness, and muscular dystrophy. This slide covers X inactivation or X chromosome dosage compensation. In females, having two X chromosomes raises the question of gene dosage. To balance gene expression between males and females, one of the X chromosomes in each female cell is randomly inactivated during early development. This inactivated X chromosome forms a structure called a Barr body, which is mostly transcriptionally silent. Inactivation ensures that the females do not overexpress X-linked genes compared to males. Sex chromosomes, like all chromosomes, can evolve over time. They can change in size, gene content, and even swap genetic material with autosomal, non-sex chromosomes. The Y chromosome in particular has experienced significant degeneration over evolutionary time, losing many genes. However, some genes on the Y chromosome are essential for male development and reproduction. Sometimes there are variations in sex chromosome composition, leading to conditions known as disorders of sex development. For example, individuals with Klinefelter syndrome have an extra X chromosome, denoted XXY, while those with Turner syndrome have only one X chromosome. DSDs can result in atypical development of sexual characteristics and may require medical intervention and support. Now let's talk about genetic linkage. This refers to the tendency of genes located on the same chromosome to be inherited together during the process of genetic recombination. Genetic linkage is a fundamental concept that was first discovered by Thomas Hunt Morgan in the early 20th century through his studies on fruit flies. So let's talk about crossing over. This phenomenon of genetic recombination, which partially breaks the linkage, occurs during meiosis. Specifically during prophase 1 of meiosis, homologous chromosomes exchange segments of genetic material in a process called crossing over or genetic recombination. Crossing over can lead to the exchange of alleles, gene variants, between homologous chromosomes, resulting in a new combination of genes on the same chromosome. Now let's compare independent assortment versus linkage. The principle of independent assortment proposed by Gregor Mendel suggests that genes located on different chromosomes segregate independently during meiosis, leading to a wide variety of gene combinations. Genetic linkage is the exception to the independent assortment because genes located on the same chromosomes tend to be inherited together rather than assorting independently. Now let's talk about recombination frequencies. Recombination frequency is the probability of crossover between two genes during meiosis. It is measured in centimorgans and is used to determine the relative distance between genes on a chromosome. Higher recombination frequencies indicate that genes are further apart on the chromosome, while lower frequencies indicate that genes are closer together. The degree of linkage between two genes on the same chromosome is quantified by the recombination frequency. This is the proportion of offspring that show a recombinant, non-parental, gene combination as a result of crossing over. A recombination frequency of 0%, as we've already said, indicates complete linkage, where 50% indicates independent assortment. So with genetic linkage, we can have linkage disequilibrium. This is a term used in population genetics to describe the non-random association of alleles at different loci, gene locations, due to their physical proximity on a chromosome. Linkage disequilibrium, LD, is a non-random association of alleles at different loci. LD is caused by a variety of reasons such as genetic drift, non-random mating, selection, mutation, and migration. Understanding linkage disequilibrium can be important in the studies of human genetics and complex traits, as it can help identify genetic variants associated with disease. Genetic linkage is the tendency of genes located on the same chromosome to be inherited together due to their physical proximity. Understanding genetic linkage is crucial for mapping genes, studying inheritance patterns, and identifying disease-causing genes, among other applications in genetics and biology. So now let's move into a new topic, non-disjunction. Non-disjunction is a genetic phenomenon that occurs during cell division, particularly in meiosis, where chromosomes fail to separate properly. It can lead to abnormal distribution of chromosomes in the resulting cells, resulting in aneuploidy, a condition characterized by an abnormal number of chromosomes. Non-disjunction plays a significant role in genetic disorders and developmental abnormalities. So let's do a quick rundown of normal chromosome segregation. Before cell division, chromosomes replicate, resulting in two sister chromatids held together by a centromere. During mitosis and meiosis, these chromatids must be properly separated into daughter cells. There are three types of non-disjunction. There is nullisomy, monosomy, and trisomy. On this slide, we'll talk about the difference between mitotic and meiotic non-disjunction. Let's start with meiotic. Meiotic non-disjunction, we'll talk about specifically in meiosis 1. So during meiosis 1, homologous chromosomes are supposed to separate. Non-disjunction in meiosis 1 occurs when homologous chromosomes fail to separate, resulting in one daughter cell with both homologs and the other with none. If a gamete with this abnormal chromosome number is involved in fertilization, it leads to trisomy, 3 copies of a chromosome, or monosomy, 1 copy of a chromosome in the offspring. Let's talk about meiotic non-disjunction in meiosis 2. So during meiosis 2, sister chromatids are supposed to separate. Non-disjunction in meiosis 2 occurs when sister chromatids fail to separate, leading to one daughter cell with both chromatids and the other one with none. If a gamete with this abnormal chromosome number is involved in the fertilization, it is typically result in disomy, 2 copies of a chromosome in the offspring. So let's talk about some of these conditions that result from non-disjunction. So non-disjunction can lead to various chromosomal disorders, including trisomy 21 and extra chromosome 21 that results in Down syndrome, trisomy 18 and extra chromosome 18, which causes Edwards syndrome, trisomy 13, which causes Patau syndrome, monosomy X, which is missing one X chromosome, results in Turner syndrome. Klinefelter syndrome is when an extra chromosome is added. Aneuploidy often leads to developmental abnormalities and intellectual disabilities. However, the severity of these conditions can vary widely depending on the specific chromosomal abnormalities. Some aneuploid conditions are incompatible with life and result in miscarriages or stillbirths. Genetic counseling and prenatal testing, such as amniocentesis or chronic villus sampling (CVS), are offered to individuals at risk of having offspring with chromosomal abnormalities due to non-disjunction. So to wrap everything together, non-disjunction is a genetic event where chromosomes fail to separate properly during cell division, which leads to aneuploidy. It can occur during mitosis or meiosis and is a significant cause of chromosomal disorders in humans, with consequences that range from mild to severe developmental abnormalities and intellectual disabilities. Understanding non-disjunction is essential for genetic counseling and prenatal care. So let's talk about the differences between primary and secondary sex determination. Primary sex determination is the initial determination of whether an individual will develop as a male or female based on their genetic composition. Secondary sex determination involves the development of secondary sexual characteristics, such as external genitalia, breast development, and the development of facial hair, which is influenced by sex hormones like testosterone and estrogen. So on this slide, we will talk about sex differentiation in embryonic development. During early embryonic development, both male and female embryos follow a similar path until the presence or absence of the SRY gene activates specific developmental pathways. In the absence of the SRY gene, the gonadal ridges develop into ovaries and the individual develops as a female. Ovary produces estrogen and progesterone. In the presence of the SRY gene, the gonadal ridges develop into testes and the individual develops as a male. So, of course, there are ethical and social implications around sex determination. Sex determination has a broader social and ethical spectrum, including issues related to gender identity, gender expression, and intersex individuals. It's important to recognize that biological sex is not solely binary and that gender identity can be independent of chromosomal or anatomical sex. So, to conclude sex determination, we know that it's a complex process influenced by genetic, hormonal, and developmental factors. While genetic sex determination, XX and XY, is the primary driver of sexual differentiation in humans, secondary sex characteristics are influenced by sex chromosomes during puberty. Understanding the intricacies of sex determination is essential not only for biology but also for discussions surrounding gender identity and diversity. Y-linked traits are traits or genetic conditions that result from mutations or variations in genes located on the Y chromosome. Y-linked traits are exclusive to males because females do not possess a Y chromosome. Y-linked traits are inherited directly from the father to the son. They are also relatively rare compared to X-linked traits. Some examples of Y-linked traits include male sex determination and maleness-associated infertility disorders. Y-linked traits are relatively uncommon in humans, and there are only a few known Y-linked traits. One example of a Y-linked trait would be hearing loss, which is a rare form of inherited deafness caused by mutations in genes on the Y chromosome. So to wrap X-linked and Y-linked traits all together, we know that there are genetic conditions or traits that result from mutations or variations in genes located on the X and Y chromosome. X-linked traits are more common and can affect both males and females, where Y-linked traits are relatively rare and exclusive to males. Understanding these inheritance patterns is important for genetic counseling and the study of genetic disorders.